Hi, my name is Thomas Skeer.
And I'm Hannah Checkeye.
And tonight we're going to be talking about lysozyme. So lysozyme is an enzyme that helps fight off bacterial cells that enter your body. It's found primarily in human white blood cells, but it can also be found in the liver, spleen, saliva, tears, and other mucosal layers inside your body. It is essentially the first line of defense when it comes to fighting off bacterial cells. So, for example, when you eat food, the lysozyme cells, enzymes that are in your mouth will immediately attack the bacteria that's on the food or that could be on your hands as you present the food to your mouth before it gets to your digestive system and can cause a bigger problem. The enzyme itself belongs to the hydrolase class of families, and we're going to go into a little bit more detail about the structure of the enzyme and how it actually attacks the cells in the next coming slides. So the structure of lysozyme, it is a globular protein that is a monomer, so it has just one single subunit. It has two main domains though. It has the alpha domain, which is from the amino acid residues 1 to 38, and then from 86 to 130, and it's made up of 4 total alpha helices. But it also has the beta domain that is located between the amino acid residues 39 to 85, and it has a significant beta sheet structure, as you can see in that picture over there on the left-hand side. It has 4 disulfide bonds that help stabilize the whole enzyme itself, and it has a pI of 9.32, an extinction coefficient of 38,940 centimeters per molar. So as you can see here, you have the mRNA sequence that is 748 base pairs long that codes for the amino acids. And on the left-hand side, you can see the amino acid sequences with the 4 disulfide bonds that help stabilize the total enzyme. So how does the lysozyme actually attack the bacterial cell? So bacteria have this tough outer cell membrane made up of carbohydrates. The lysozyme essentially just attaches to the carbohydrate chain that surrounds the genome of the bacteria. It affects that chain, and then after damaging it, the bacterial cell essentially explodes due to the pressure difference from inside the cell compared to outside of the cell. So as you can see here, this is a little bit better picture of what I just explained. The lysozyme right here is attaching to the carbohydrate chain, which is located right there. The lysozyme cleaves at the beta 1 to 4 glycosidic linkages between N-acetylmurmic acid and N-acetylglycosamine of the carbohydrate chain. Like I said before, after this chain is compromised, the cell will actually explode due to the pressure difference. Now, some researchers do believe that the smaller the bacterial cell, the less number of glycosidic linkages the lysozyme actually has to affect. So for smaller bacterial cells, it could need between 2 and 4 linkages damaged, but for larger ones, it could require as many as 10 linkages to be damaged before the cell will actually explode. So here we go. What inhibits lysozyme? There are many different things that can cause lysozyme to not to function as well as it should. One of those being sodium dodecyl sulfate. So sodium dodecyl sulfate acts as a way to stabilize the lysozyme enzyme. It's very small, so it's able to sort of maneuver its way into the interior of it, and it essentially just locks down the molecule into its conformation. Urea is also a catastrophic agent, so it can cause the protein to denature. Because of their very small and highly polar electrostatic abilities, it will break up the protein. Alcohols are widely used as protein denaturants across all of biochemistry. They have the ability to cause precipitants and crystallizations along with the denaturing process. The hydrophobic sites on the enzyme's surface have the ability to absorb the alcohol molecule, which then reduces the attractive hydrophobic protein-to-protein interactions that the lysozyme requires to attach to the carbohydrate chain. So then finally, oxidizing agents. The lysozyme has extensively been studied for its ability to refold and fold consistently. However, oxidizing agents, however, have a strong enough effect to actually stop the lysozyme from refolding properly and compromises the integrity of the structure itself. So separation strategies. One way to separate lysozyme from other different enzymes is to use a cation chromatography exchange. The separation of lysozyme can be done Once the lysozyme is trapped in a glycine sodium hydroxide buffer at a pH of 9.3, the elutin is then desalted and then ultrafiltrated to really isolate the lysozyme. In a study from Iowa State University in 2016, lysozyme was actually separated from an egg white, and then it was used to then help immunocompromised chickens. It was reimplanted into immunocompromised chickens to help them develop an immune system.
So for our primary literature source, we looked at a study called The Analysis of the Native Structure, Stability, and Aggregation of Biotinylated Human Lysozyme. So in 2012, 7 scientists, they're listed on the works cited page, had a goal to find a way to fluorescently label the mature fibrils of the human form of the lysozyme without actually changing the native structure of the lysozyme, so that they could monitor and understand the process by which aggregation occurs in the lysozyme. In the past, many studies have examined the folding pathway of the lysozyme, and they discovered 2 natural variants that have actually been found to be linked to systemic amyloidosis. And systemic amyloidosis is a rare and also fatal autosomal dominant disorder that's caused by the fibril deposits in the kidneys, liver, and spleen of the lysozyme. So by finding a way to label the human wild-type lysozyme without actually ruining the native structure, they hope to be able to monitor the in vitro pathway through which lysosome aggregation actually occurs and then causes systemic amyloidosis. So in the methods, the first step they did to label the lysozyme, they took the wild type and they biotin labeled it using N plus biotinyl 6 aminocaproic acid and succinimidyl ester, or short bio-NFE. And the biotin labeled lysozyme was then purified using a tris buffer and a Capto-S ion exchange column before it was eluted through a sodium chloride gradient in order to collect multiple fractions. Then they did a circular dichrosum spectroscopy using the JASCO J810 spectropolarimeter, and experimental data was then graphed and put it into a sigmoidal curve using the melting temperature and the fraction of the unfolded protein for the X and Y axes. Then the next step was to take a nuclear magnetic resonance spectroscopy using the Bruker Advanced 700 MHz NMR spectrometer. The samples were run on an SDS page gel, they were reduced to DTT, and then alkylated by iodoacetamide, digested with trypsin, and then loaded into an autosampler for analysis. Lastly, aggregation studies were performed in 0.1 molar sodium citrate buffer at a pH of 5.0 at 60 degrees Celsius in the presence of a 3 molar urea, which we talked about earlier as a chemical detergent. So, results, the researchers were actually able to successfully biotin label the specific lysine 33 residue of the wild-type lysozyme, while still maintaining very similar native state properties of the wild type of the lysozyme. So after doing a number of biophysical techniques to compare the wild type lysozyme to the newly biotin labeled protein, they concluded that the secondary structure of the labeled protein was almost identical to that of the wild type. So the respective thermal stabilities were the melting temperature of 76.0 plus or minus 0.5 degrees Celsius for the wild type lysozyme, and 75.9 plus or minus 0.5 degrees Celsius for the biotin labeled lysozyme. The biotin labeled enzyme also retained about 60% of the enzymatic activity of that of the wild type. Additionally, both the proteins were observed to still form aggregated fibrils, and they also both displayed sigmoidal curves typical of the wild type aggregation pathway. Aggregation has been determined in the past to occur in vitro in the wild type under specific conditions, specifically elevated temperatures like the 60 degrees Celsius they used for this method, as well as in a low pH and in the presence of chemical detergents in solution. So this is why they use the 60 degrees Celsius in the 0.1 molar citrate buffer at a pH of 5 with the 3 molar urea. Aggregation occurred after a 4-hour lag phase when going through this process. And then lastly, transmission electron microscopy images also confirmed that the size and structure of the fibrils that were formed by the biotin-labeled protein were essentially identical to the fibrils that would form in the wild-type lysozyme. So as you can see here were some of the images and results that they found. So in the first one, you can see up here, this is the wild-type lysozyme, its molecular weight. As well as down here, you can see the biotin-labeled enzyme. They're very, very similar. And then over here in picture B, this is actually the primary sequence of the human lysozyme. And it shows the site where trypsin digestion occurred. So in the biotin-labeled enzyme, it's the open gray arrows. And then for the wild type, it is the filled black arrows. And you can see that specifically at residue 33, that is the only site where trypsin digestion does not occur in the biotin-labeled enzyme. And then in C, you can see the secondary structure of the labeled lysosome denoted by the dashed gray line, and as well as the secondary structure of the wild-type lysozyme with the solid black line. And they used FAR UV-CD for this method. Lastly, this is the protein unfolding curve that we talked about before. So these open circles are the biotin-labeled protein, and the wild type are the black squares. And once again, you can see it follows that sigmoidal curve, and the melting temperature where 50% of the protein is unfolded are very, very similar. And then in this picture, you can see the fluorescence of the lysine residue 33, right around here. It has the highest peak, as well as in this picture of the structure of the lysozyme, you can see right here where the location of the lysine 33 residue is actually found. And then in this picture, this shows the in situ binding fluorescence kinetics of, again, the biotin labeled in the gray and the wild type in the black. Again, very, very similar. This was in the presence of the 3 molar urea in that 0.1 molar buffer pH 5.0 with constant stirring at the 60 degrees Celsius. These are transmission electron microscopic pictures of the endpoint samples of the aggregation reactions. So you can actually see the fibrils, the aggregated fibrils formed in the wild type and again in the biotin labeled protein. This is pretty similar in this picture. It's a dot blot assay of the fibrils that are formed by the wild type and the biotin type. And then over here we have the binding fluorescence kinetics of the wild type in the absence and also in the presence of 10% bio-labeled fibril seeds. And then lastly over here we have the conformational stability of the biotin labeled in the circles and the wild type labeled in the squares. So through all of these pictures we could see that they were successfully able to label the lysozyme in order to in vitro watch that aggregation pathway in hopes for future studies to be able to kind of figure out what happens and how this process causes systemic amyloidosis in hopes of finding a future cure. So thank you for listening, and here are our, we're excited.
Thank you.